All right, welcome back. So I have the pleasure to introduce Professor Prerna Sharma from IISc. And Prerna is going to talk about enhanced mixing and vortical flow around confined microalgae. Over to you, Prerna.
Good afternoon, everyone. I'm going to talk about mixing and flows around a particular algae called Chlamydomonas. So the work that I'm going to show you has been carried out by two of my talented PhD students. They were Smita and Sujit. And the theory collaborations were with Professor Shriram and Professor Aparna. And so on the right, you're seeing this picture where this round bright object is the algae. And these beautiful streak lines are actually the trajectories of tracer particles that we had seeded in. So this particle, this cell is moving through the fluid, and it's setting up these vortical flows around it. And as you can see, as you go further away, the trajectories are becoming more Brownian-like. So I'm going to talk to you about what happens to these vortices as you confine the cell physically. So the outline of my talk is that I'll introduce this model organism called Chlamydomonas NHT. Then I'll talk about the role of confinement in Chlamydomonas flow fields. And then if time permits, we'll also discuss a little bit about the phototaxis that is exhibited by this organism and collective effects in that, and then conclude. And so essentially, Chlamydomonas reinhardti is an example of a self-propelled active particle, or it's often modeled as an active Brownian particle that Thomas talked about in the morning. And the reason it's good to study this system is because it has also ecological relevance. It's involved in these large-scale blooms that form, and it's also is used in industrial bioreactors to produce enzymes and drugs that are of benefit to humankind. And so this is how the cell looks like. Here we are holding the cell on a glass pipette, and so since the cell is sort of held on a pipette, you can see how its flagella beat. So its flagella beat in a breaststroke manner. The flagella beat frequency is about 50 hertz, and the cell speed, if the cell was not mounted on the pipette, it would run at 100 microns per second. So that's a very fast speed. And if the cell is free swimming, then this is how it moves. So essentially, this synchronous motion of the flagella enables the cell to move forward. This organism is considered a puller type of swimmer, in the sense that it pulls fluid from back and front and pushes the fluid out from the sides. Essentially, you want to ensure divergence of V0. And this kind of picture, simplistic picture, holds true in the far field. If experimentally, when you measure the flow field of this cell in unconfined fluids, you find all the structure. And so essentially, if you look at the far field limit, you can see that the fluid is being pulled from the front and back and pushed out from the sides. So how do you theoretically understand this? The way this classical study analyzed it is by saying that we'll have 3 Stokes-Slit. So Stokes-Slit is like a point force in the fluid. And the 2 Stokes-Slits, the green Stokes-Slits are placed at the average flagellar position. And there is a red Stokes-Slit which indicates the hydrodynamic drag force that the cell experiences. And so by having these 3 Stokes-Slits, you can nearly capture all the signatures present in the flow field. So that's the story, in unconfined fluids. Now, why should we really think about confined geometries? The reason is that this organism, even though we grow it in the lab in, big liquid cultures, and It's a very easy organism to work with. You can grow it in millions, and large collection of mutants is available commercially. So, so confinement is important because this organism naturally encounters extremely confined environments. It's one of its habitat is soil, rocks, and pools. And in lab experiments, of course, people would look at them in microfluidic channels, in slide cover slip geometry. And more recently, there is some technological implication that if you really squish these cells, then you can get upregulation of lipid synthesis. And so it has implications for biomass production. And so we can ask what happens to the motility and flow fields of algae under strong confinement. So flow fields under confinement, there is a theoretical expectation for that, which is that the flow field will resemble that of a 2D source dipole that's oriented along the swimming direction. So the cell is swimming to the right, and you get this just two-loop force field, two-loop flow field, and that is the theoretical expectation, which only accounts for hydrodynamics. So it basically says that you have two boundaries, and so the flow velocity has to go to 0 at the boundary, and with this boundary condition, how will the flow fields change? When we did the experiments, we found something different, and so we basically measured simultaneously the motility and the flow fields. And the force balance that you would expect is that in a weak confinement, so the cell diameter is about 10 micron. So we prepared these chambers where the height of the chamber was about 30 micron, and the cell was unconfined in the XY plane. So there's confinement only in one direction. And here it behaves more or less like the bulk, where the thrust forces of the flagella balance the hydrodynamic drag experienced by the cell body. Now, in the case of strong confinement, when the chamber depth is comparable to the cell diameter, then there is an additional contact friction that the cell experiences. And it is the contact friction that the previous theoretical studies missed including in. And so now the force balance is the thrust force of the flagella has to not only overcome the hydrodynamic drag but also the contact friction. And basically the contact friction dominates essentially, as I will show you. And there is some variation in the average cell size. So one has to think about the degree of confinement, which is the ratio of the cell diameter to the height of the chamber. I'm going to plot the data as a function of this ratio. So here is a movie of the cell in 30 micron chamber. These black dots are the tracer particles. And I think the resolution is poor, mainly maybe because we are on Zoom. And then, in the extremely confined regime, you can see that the beads are actually tracing out vortices. So the flow becomes enhanced. And there is another class of motility that shows up in the extremely confined regime, where instead of getting these beautiful synchronous flagellar movement, you get this paddling movement of the flagella. So here basically the flagella itself is undergoing contact interactions with the wall, and we do not study these cells, or we do not include them in our analysis, because really there is no way to have properly averaged flow fields. And so the two kinds of behaviors, this wobbler versus synchronous, is mainly a function of cell diameter. So when the cell diameter is exactly the same as the chamber height or larger than that, then you get this really synchronous movement of the cell. And below that you get the paddlers or the wobblers. And that's mainly happening because essentially when the cell size is less than the chamber diameter, then the cell cell itself tries to have a helical trajectory. And so now the cell body is able to rotate, and that's kind of enabling the flagella to interact with the walls. Whereas in the synchronous case, you have really tightly squished the cell, and the flagella are just beating in plane, and the flagella are not really seeing the boundaries. Only the cell body sees the boundaries.
Cell diameter by.
We don't control the cell diameter. We grow up a culture. There is a heterogeneity in
Okay.
cell size, and you just observe it. Yes.
Micron height,
Yes.
that is also considered confinement. Are you saying that's unconfined there?
That's practically unconfined.
So you're not restricting one other degree of freedom of motion, or
Yes, so
when you say helical motion, what do you mean by that?
Okay, so that's a detail that I did not share. So essentially, in addition to the flagellar beat, the cell body also rotates about its own axis. So you have, so the cell is, and then the flagellar beat itself is noisy. So, it's not always perfectly breaststroke. It will sometimes have phase slips. So there is yaw as well as roll of the cell body. So when you combine yaw and roll, you get helical trajectories with, very large radius of helix. So usually you wouldn't worry about it, but when you confine, then, the cell is, really running into the boundary essentially. Yeah.
Excuse me.
Yes. Question.
Ma'am, you are
Yes.
yeah. So from 12 micrometer to 14 micrometer,
Yes, yes.
it's showing synchronous motion.
Yes, yes.
So you are saying because in that regime it is confined.
It is tightly confined.
Tightly confined.
Yes.
So are you also moving your cover slip as the cell diameter is increasing?
No, our chamber is fixed. We made the chamber with, these double-sided tapes of fixed thickness.
Okay.
So we have a culture where we have a variation in cell size. We inject the culture into the chamber, and then we observe them. And
So I'm asking, at 12 micrometers also it's tightly confined.
Yes.
At 14 micrometer also it's tightly confined.
Yes.
But the chamber size remains same.
Yes. Chamber size is fixed. So essentially, all this plot is trying to say is that you will observe synchronous behavior for cells with size between 10 to 14 micron. The chamber is fixed. In the same chamber, if you introduce a suspension where the cell size varies from 8 to 14, any cell whose diameter is between 8 to 10 will fall into the wobbler category, and between 10 to 14 will fall into the synchronous category.
What are the dimensions of the chamber?
10 microns, exactly.
Okay.
Yeah. So that's why the boundary is at 10 microns. Yeah.
Okay. Thank you.
Yeah, and so you can look at the trajectories of these cells. So for clarity, we have, put the starting point of all the trajectories at 0, 0. And the top versions are at zoomed out, and the bottom versions are zoomed in of the same set of trajectories. So what you can see is that in this H30, which is the weakly confined regime, 30 micron chamber, the trajectories are nearly straight. And if you zoom in, you see this kind of zigzag. And this zigzag comes from, yes.
I see you go up to almost 1.6 millimeters. So is it one field of view with a very low magnification?
Yeah.
Or okay. That's what I said. Or
Yeah.
I was going to ask, do you have a tracking microscope?
No, I don't have a tracking microscope. Yeah. I would love to build one though. Yeah. I mean, it's easy to build. It's just that I haven't sit down and made it. So so you see this zigzag pattern in the trajectories, and that's characteristic of this swimmer. So this swimmer has a power stroke and a power stroke where the flagella kind of flap down, and then a recovery stroke where the flagella go back up. And so the way this swimmer effectively moves is that it will go forward in the power stroke, and then it will go slightly back in the recovery stroke. And the forward and backward are not equal amounts, so that's why the organism is able to move forward overall. And then in the wobbler, you see that the trajectories become more loopy and ill-defined. And then in synchronous, it's extremely loopy, and you will see that the cell centroid is just going, is spanning out these very tightly wound circles. So you can characterize the nature of these trajectories by measuring what is called tortuosity. So tortuosity is the ratio of the arc length distance between 2 points divided by the shortest distance between 2 points for a given, I think we chose some 10-beat periods. And then you can also plot the cell speed. So what you find is that as you increase the confinement, the cell speed comes down dramatically. So not only these cells are going in tight circles, they're also much slower than the weakly confined swimmers. And the tortuosity goes up. So the cell speed decreases dramatically, but the cell doesn't really stop. It's just moving extremely slowly. And so here is the unconfined flow field by these authors. And then in a partially confined regime, this is what we found. So it's very similar to the unconfined flow field, except that these outer vortices have sort of, become closer, and the stagnation point is also closer to the cell body. But otherwise, the direction of vortices, everything else seems very similar. In the strongly confined regime, you only get 2-loop flow field. The stagnation point is now even closer to the cell body. And most importantly, the vortices have changed direction. So here the vortex was anti-clockwise, here it's clockwise. And so there is an inversion of the vortices that we are seeing, or there's an apparent inversion. One intuitive way to understand what's happening is that these vortices are actually moving in, and it is these vortices, that have the opposite handedness. And these vortices have basically become much more, closer to the cell body for us to resolve. So, but anyhow, how do you theoretically explain what is going on? Because the conventional expectation was to get just 2 loops with the opposite sign. So, yeah, so this is the comparison with the theoretical prediction. You could do many things. You could say, okay, I'll put 3 Stokes slits, but yes.
So this is still in water, right?
Yeah,
you haven't increased the viscosity or anything?
No.
Okay.
Yeah, everything is in buffer. Yeah, aqueous buffer. Yeah.
Okay.
Yeah, and so, you could say, yes, sorry. Mic is on.
Will you go to previous slide?
Yeah, sure. Here. Yes.
Actually, I don't understand the trajectories of flow profile, how it is getting changed from second figure to third figure. I mean, the theoretical understanding or like anything which I can.
Yeah. I mean, that's what my talk is about, how you will get that. But all I was trying to motivate is that, look, how do you go, how would you go from here to here? Is that you will shrink these vortices, make them very close to the cell body. And then you move these vortices here. So if you move these vortices here, they will look like that. The signs would look like that. Okay,
ma'am. Thank you.
Yes. Yes.
My question is, like, under the strong confinement, the beating frequencies and all, they remain constant?
Yes, beating frequency remains constant. Yes. Because the flagella is really not seeing it's not going contact interactions with the wall. So the beat frequency remains unchanged. Yeah. So you could ask, one way to theoretically probe this would be to say, okay, we'll still place a 3-Stokes slit, and we will just account for the boundary conditions of, no flow at the walls, and see what happens, and you get this very complicated flow field. Or you can do the source dipole calculation, and both of them would really not match with what is happening in the experiment. So then how do you go about addressing this? Or what's the origin of the flow field inversion? And so we go back to this force diagram that I had initially discussed, where in the weak confinement, the thrust forces balance out the hydrodynamic force. So the hydrodynamic force is 3 pi eta d times u. And in the strong confinement, essentially, the speed is coming down. So the drag is much smaller than here, and you have this extra contact friction. So now mostly the flagellar thrust has to work against the contact friction. Okay, and so it is this change in the nature of force balance that's driving the vortex flow inversion. And essentially, because we are only measuring the flow fields, we are not really measuring the contact friction. All I need to do is I need to place confined Stokes slits at the 2 flagellar position. The hydrodynamic drag is negligible, and so I wouldn't worry about it. And that should give me the experimental flow field. So yes.
C.
Yes.
You are saying that H is less than or equal to D. D is the diameter of the flagella, right?
D is the diameter of the cell body. Yes.
Higher diameter than the width of 2.
Yeah, so that's the experimental design, right? So as an experimentalist, I know what's the spread in the cell size. It's 8 to 14 microns. And so I designed my chamber to be exactly 10 microns.
Okay.
And so then, of course, I'll always find some cells whose diameter is larger than or equal to the spacing between the 2 plates.
Okay.
Yeah, I mean, yeah, cell is squishy.
Okay.
Yeah, you can just like squish it a
Okay,
little bit.
thank you.
Yeah, we tortured the cells to get all this. Yeah, that's the concern. Yeah, I mean, it's not silica or something which you can't. Yeah, so then we'll just quickly go through the nuts and bolts of how you would calculate the flow fields. So to calculate the flow field, you need to solve the Stokes equation. And so you have this, classical Stokes equation with the continuity equation. And then you use this 2D approximation. So your chamber is, effectively giving you parabolic flow. So you can have only the first Fourier mode of the speed or the velocity. You plug it back into the Stokes equation along with the continuity equation. This becomes your equation to solve. And essentially, so this is the equation that, so here G of R is your forcing term. So if you were looking at Stokes slit, this would be like a point force. And so, if you want to get the flow field of a Stokes slit, you will put a point force and solve it to get the flow velocity. In our case, we know that the flagella of the cell are an extended object, and they're deformable. And so instead of a point forcing, we are putting a Gaussian forcing. And that lets us essentially solve it in the Fourier space much more easily. And eventually you just get a flow field corresponding to this diffuse Stokes slit that is confined. And that is confined with this boundary condition, and you invert it to get the flow field. So here you're getting it in the Fourier space, and then you invert it. So once you do that, then this is your experimental flow field. You solve the this equation which is called, with all these approximations, the Stokes equation is now called the quasi 2D Brinkman equation. And you place the two Stokes slits at the average flagellar position with the diffuse forcing term, and you get a flow field which is remarkably similar to the experimental flow field, and you get quantitative agreement. So here I am plotting normalized flow velocity as a function of polar angle for representative radial distances from the cell center, and you get a really good match. So essentially the insight was that you do not want to place all the 3 Stokes slit, you want to place only 2 Stokes slit, because they are essentially, there is no hydrodynamic drag. They are mainly working against the contact friction, which does not show up in the fluid itself. And the simple-minded source dipole picture won't work because it misses this contact friction insight. And so the coupling between the cell motility and the strong confinement alters the force generators and reduces a force dipole swimmer to a force monopole one. So what is the ecological relevance of all of this? Why, is this even really important? There is some relevance in the context of mixing. And so what we did is we looked at the velocity correlation function of these flow fields, and we found that there is slight difference in the decay of this correlation function between the weakly confined to strongly confined. The correlation length is larger for the strongly confined regime. So long range flow correlations. And then what was interesting is to look at this data slightly differently, where we plot the maximum displacement of tracer particles for 10 beat cycles of the flagella, and as the cell goes through the field of view in the weakly confined regime, and you put a color bar to these tracer displacements. So what you find is that, of course, the tracers that are closer to the cell move much more, and as you go away, they become more Brownian-like. And then, the radius of influence of this freely moving cell is actually much less than compared to the strongly confined cell, where it only just slightly rotated in that time, but had a much larger radius of influence and much larger tracer displacements. And so you can plot the mean squared, so the radius of influence was almost twice that of the weakly confined regime. And you can plot the mean squared displacement of the tracer particles between the two regimes. You can see that it becomes much more super diffusive in the strongly confined regime. So with this, we can I can conclude this part of the story, where the idea is that the strong contact friction from the walls reduces the cell speed, which then leads to inversion of the lateral flow vorticity. And the coupling between the motility and confinement alters the forces generating the flow. And that the overall picture is that all of this leads to enhanced fluid mixing despite higher friction experienced by the organism. So this concludes this part of the story. And now I will quickly show some data on phototaxis, and then we'll conclude.
Excuse me.
Yes.
Maybe a naive question.
Yes, please.
Ma'am, when you're talking about influence of radius,
Yes.
That we are talking along the length of the confinement, not along its height, right? Because
yeah, I'm talking about in the XY space. Yeah.
Okay.
Laterally. Yeah.
Okay. Yeah.
One more question. Yeah.
Like suppose the confinement is along this direction.
Yes.
And so the approximation to the flow as a 2D, like as a pipe flow kind of
Correct,
thing,
correct.
is along this direction, right?
Right.
But
right.
then you also have the boundary condition on the surface of the, algae as a
correct.
no slip.
Right.
Is it?
Right. Yes, yes, we do.
So, I mean, would that parabolic flow condition still be valid?
Yeah, I wouldn't worry about it because the flow is set up by the flagella, and I mean, the cell body is just basically being dragged along due to flagellar motions.
So it's kind of like an external, flagella is generating an external flow kind of thing with two no-slip valves.
Correct. Yeah.
Plane that you are taking, that is the mid plane of your chamber, right?
Yes.
So the tracer bead you are taking, won't they just sediment on the bottom of the cover slip?
These are 200 nanometer tracers.
Okay.
So they're extremely small. They have been.
But they will do this motion. I mean, they can go up, they can go down. So.
Sure. So basically, we have some finite depth of field.
Yeah.
So we are doing some kind of time averaging, spatial averaging anyway. So
do you track those tracer particles?
Yeah, the flow fields are extracted from by tracking these tracer particles.
So when the particles go out of your field, so you can.
Them. Huh? We forget about them because some other tracer particle will come into our field of view.
Okay, so you have a lot of.
Yeah, we have. I mean, unfortunately, the video resolution went down when we projected through Zoom.
So, but if you want to get the exact flow field, you should have some optimal density of your tracer bead.
Yes, we optimize that. Yeah.
Okay. So.
I mean, you have to do that. Yeah.
Okay, thank you.
Yes. Yeah. So actually, it's just a terminology we used that we call them synchronous swimmers. The cell naturally goes through periods of synchrony and asynchrony. Like if it's an unconfined fluid, the flagella will synchronize for a long time, then suddenly through noise leave, have a phase slip and then gain synchrony again. When we observed these strongly confined cells, they were mostly just having a synchronous beat. So that's why we call them synchronous swimmers, nothing else. Okay. Yeah, yes, go ahead.
So I just have a quick question. Why does the radius of influence, it's not immediately obvious to me why it's larger for confined cells.
Yeah, I mean, so it's not obvious why it's like this. How do I mean, this is my data, right? So essentially we, so what I can say is that, this swimmer is, has this very, it moves through the fluid very quickly, okay? So its cell speed is like 100 microns per second. So the tracers would move and then very quickly the cell is far away from this tracer. So it has no way of feeling more, kicks. Whereas here, the cell is moving much more slowly and it's just going in circles. So these tracer particles continue to feel the flagellar beat and have a larger displacement. I hope that sort of explains. Yeah. Yes, you had a question?
Yeah. So across the two panels, because in one case the flagella, I'm assuming that the white dashed circle is the extent of the flagella.
No. The white dashed circle is the radius of the influence.
Okay.
I think we set off the cutoff at orange somewhere, and so with the same cutoff, the radius of influence happens here, and for this it happens here.
And so the height is about 10 micrometer, right? So effectively the chlamydomonas is squeezed somewhat.
Yes.
So does that somehow make it more pancakey shape, and that makes the influence, length of influence?
Yeah, I mean the shape, if you look, I mean I don't have the image here, it's hidden somewhere here. The shape doesn't look a whole lot different. Yeah, I'm sure there is some squeezing happening, but not very evident. Yeah. That's why I didn't worry about the torture to the cells. It just, they visually they appeared very similar. Yeah. But the point is that, Ideally, what you would like to do is you image the cell under this extreme confinement, and then you image the same cell in unconfined fluid. Then maybe you can compare what is the original size. Visually, otherwise they look fine.
But it should change the shape of the cell, like from, let's say, if it's spherical, then it should go into elliptical or something.
It's already elliptical, and as far we couldn't tell a whole lot about the cell morphology. There's anyway a lot of variation in morphology also. So yeah, as far as we could tell, we couldn't see much difference to characterize it quantitatively. Mostly we worked with cells which were like 10 to 11 microns. We wouldn't work with cells which are like 14 microns, because then those are extremely squeezed. Yeah, so most people often ask me, okay, you have looked at 30 micron, you have looked at 10 micron. How do you go between these two regimes? And can you continuously change the height of the chamber, etc.? And yeah, there's only so much we could do, but I think Tapa had an interesting suggestion why that could be an good experiment to look at. So yeah, so I'm going to talk to you about the next part where we look at how does this organism respond to light. So these so algae cells run towards light, blue light. Basically, they're like little plants. They want to do photosynthesis, so they want light. And other microorganisms like sperm would do chemotaxis. They will run towards the egg in response to chemical gradients. And so this sort of directed motion of cells, it empowers the organism to reach favorable conditions for thriving. And traditionally, biologists have looked at what this behavior of taxis on single isolated cells. And that's important because that tells you the mechanism, how does the cell sense and respond. But we also know that a cell swims differently when it is part of a dense suspension compared to when it is in isolation. So how does the swimming, sensing, and response couple at the collective population level? And so we wanted to fill in the gap for collective phototaxis. So I'll show you, so here now these black dots are individual cells. We are looking at low magnification, and these cells in absence of phototactic light. So when I say it's pitch dark, I mean absence of phototactic light. We are imaging them in infrared light or red light where they're not very responsive. And so you will see that in pitch dark, sorry, I didn't mean to. In pitch dark, the cells are just running in random directions. So it's a persistent random walk. Okay. Now when you turn on the blue light, they will all run towards the top. But if you notice carefully, you will see that some of the cells are running in the wrong direction. And so the question is, what happens to this fraction of incorrectly moving cells as you change the cell concentration? So how does Clemmy scan its surroundings for light, or how does it detect? So it has a primitive version of eye called an eye spot. These proteins are responsive to blue light. And essentially what happens is that when light falls on the eye spot, it causes downstream biochemical changes that then change the motility of the two flagella and reorient the cell towards the light. So the flagella closer to the eye spot is called cis, and away from the eye spot is called trans. And so it's, as I had mentioned, this cell it has this slightly non-planar flagellar. So it has this slight asymmetry in the beating of the flagella, and then it rotates its cell body about its own axis. So these two combine give it a non-planar flagellar beat and let it span a helix. So if you had only asymmetry in the beating of the flagella, the cell will go in circles. If you had only spinning about the body axis, the cell would go straight. The combination of the two gives you the helical motion. And you can visualize this The eye spot becomes bright under polarization microscopy. And so this bright thing is the eye spot. And then you can just watch the cell, you can see that the body is spinning. The eye spot goes from left to right. Okay. And so essentially by spinning the body, I am scanning, the cell is scanning the 3D environment for presence of light stimulus. And then what happens when the light stimulus falls? As soon as the light falls on the eye spot, then the beating of the flagella closer to the eye spot, the cis flagella is inhibited and the trans flagella is enhanced. And so that turns the cell towards the light. And this, and then when the eye spot is away from the light, then the reverse happens. And so eventually the cell just continuously turns towards the light. So this mechanism was found out by holding the cell, oops, I'm sorry, I didn't mean this, by holding the cell on a pipette and watching what happens to the flagellar beat as you shine light. This was done in a classical study in 90s where, you know, we didn't have high-speed cameras, and so this sort of imaging was done stroboscopically. Now with modern tools of high-speed microscopy, you can really visualize what happens. In absence of light, the two flagella are beating symmetrically, but in presence of blue light, the beating of this flagella is suppressed and the beating of this flagella is enhanced. So if this cell was not held on a pipette, it would just turn towards light. So most studies of this, studies related to mechanism of phototaxis have been done on a single cell. So the free swimming behavior is missing. And the, yes.
But anyway.
Yeah, now it's
great. So which flagella moves more? Does it depend on the angle of light or the intensity of light, or how you're applying the light by any chance?
Yeah, so it depends on.
Because eye spot is like.
Eye spot is always rotating.
Always rotating.
Yeah, yeah.
So why doesn't it actually change the which flagella?
Yeah, it does switch it. So that's this mechanism, right? So for example, this is the light, eye spot is here. So this is suppressed, this is enhanced. When the eye spot goes here, then this is suppressed and this one is enhanced. So the closer, so the eye spot is here now in the shaded regime. So the one that's closer to the eye spot is now being enhanced and the one away is suppressed. Yeah. Whole body rotates. Yes. Yes, yes, yes. Correct. Yes. So that's what this panel is showing. So here the cell is on the pipette. In absence of light, the two flagella are beating the same way. In presence of blue light, eye spot is here, this one is suppressed and this one is enhanced. And that will just continue as long as the light is on. Because the cell is fixed, the light is fixed, the eye spot is fixed. So, yeah.
There are three kinds of motion. One is the direct and other one is getting back reflected, and third one is the like changing path spherically or helical motion kind of things.
It
Right?
is only
if you go to the previous slide.
Yeah.
You
I
are
was just
mentioning
trying to
there are-
explain how the helical motion arises.
There are three kinds of path if you see.
There are three possibilities,
Okay.
depending on how the flagella were beating.
Okay.
So if you did not have the spinning of the body axis and you just had slight asymmetry, then the cell will go in circles. But in reality, it doesn't.
Okay.
Because its body is also spinning.
Okay.
So when you combine these 2 degrees of freedom, you get helical.
Okay.
Yeah.
So like at the end, it will go in a helical motion, right?
It will always go in helical motion.
Okay. I thought maybe there are three kinds of path and they are all. Okay. Okay. Thank you.
Now, if you increase the wavelength of this helix and make the radius of the helix very tight, it may appear the cell is going in a straight line, but there's always a 3D motion to the cell. Okay. So what is known about the collective phototaxis of cells? So, I mean, I was not kind to the biologists. Actually, they did observe collective phototaxis in the sense that these are classical experiments where you have the stimulus light coming from the top and you have a cell suspension here. You have a beam to measure the optical density. So what these people found out was that so essentially when you turn on the light, the optical density will keep on increasing. So this rate of phototaxis, which is the change in OD per minute, is a function of cell concentration. So this was, so it was established that there is collective phototaxis. This mechanism is unknown. Individual cells were not tracked. More recent studies have done cell tracking and found the same thing. There is some critical concentration above which the efficiency of the phototaxis increases. The mechanism remains unknown. And so we wanted to understand the mechanism of the collective phototaxis and use modern tools of high-speed imaging, single cell tracking, things like that, and also look at kinetics of turning and so on. Our experimental setup was simple-minded. I have this chamber containing cells. I am looking from below at low magnification. The stimulus light is at one end of the cover slip. The cells are very sensitive to light. So, even though light is like far away, they can really phototax very well. And we image the cells. And so again, these movies. So first you characterize the motion in the dark. So you will measure what is called mean squared angular displacement as a function of time to get the rotational diffusion coefficient, which is the T rot that Thomas was talking about. And so T rot will be inverse of this rotational diffusion coefficient. The rotational diffusion coefficient is independent of cell concentration in our case. And so that sets the, the background of how these cells are behaving in the dark. Now, in the presence of light, as I said, the cells run towards the light. You can, again, plot the trajectories. These trajectories are colored arbitrarily just for visual clarity. And you can ask what fraction of trajectories are within some 10-15 degrees of the light source. So sorry, this is confusing. In the trajectory plot, the light is here. Whereas in the video plot, the light is here. So most of the cells are running towards the light. And this efficiency increases with light intensity, as you would expect that if there is more light, the cells would do much better. And you can plot the probability density of these trajectories as a function of angle. And so black is when it is pitch dark, so all directions are equally probable. And then directionality picks up as you increase the intensity. So here is the collective phototaxis data. Again, the light is from below at 0, 0 here. As you increase the concentration, you find that initially all the cells are going towards the light. Then at some intermediate concentration, there is confusion. A large number of cells are going in the wrong direction. And then as you increase the concentration again, then most of the trajectories are again aligned along the light. And so if you were to plot the probability density, you will see that initially the peak goes down and then it goes back up as you increase the concentration. I have shifted these plots for clarity. The light is always in one direction. And you can plot the phototactic efficiency and you will see the similar behavior that.
Prerna?
Yes.
So as you increase the concentration,
yes,
the highest concentration, it seems like there is a collective motion towards the left or something. So it starting from the 0, it doesn't quite follow a vertical motion towards the light. Is it because the collective swimming has a drift motion that happens?
Possibly, but see, because we are measuring all these efficiencies within plus minus some 15 degrees, it's fine. And statistically, I'm not sure if this feature is very reproducible. I mean, we have taken like, I mean, already this data set has millions of trajectories in it, but we have done. Yeah. Yeah, I am not sure if I would take that seriously as of now. Because, you know, here they are fine, and I'm sure there are plots in between which were fine and so on. Yeah. And so, so at low light intensities, you can see that the efficiency goes down and then goes back up. At higher intensity, you have still similar pattern, but much more milder, as you would again expect. And so I'm not going to tell you why the efficiency goes down. I have a story, or rather hypothesis and evidence as to why the efficiency would go up above this critical concentration. So, and later on we did this experiment for large number of light intensities and cell concentrations, and similar reentrant behavior was seen. So, here is the active Brownian particle model. So, essentially a cell running towards light is an active Brownian particle in presence of an external potential. And so here the light is like effectively an external torque that will reorient the cell towards light. So our potential is going to be angle dependent, and then you can write these equations of motion, and then go back to the Fokker-Planck picture of probability density, solve it to get the expected distribution function for the angular probability density. So this is called a von Mises distribution. If you plot it, it will look like a Gaussian. And it fits well to the data. So the fitting parameter is the ratio of the strength of the torque to the rotational diffusion coefficient. So because we know the rotational diffusion coefficient, we can extract the torque. And as I had mentioned to you, the rotational diffusion coefficient is independent of the cell concentration in the range we explored. So essentially we have a cell density dependent torque, and that torque increases as you increase the concentration above the critical concentration. So this simple-minded theoretical picture or phenomenological picture works. Now the question is, why do we have this kind of behavior? So to address that, we looked at, we said, okay, maybe the cells are, secreting some chemicals and enabling other cells to follow them, etc. Those sorts of mechanisms we couldn't really establish. The data seemed independent of that. And then we looked at physical means, where we looked at the speed of the cells. And so the speed of the cell goes down as you increase the concentration above the critical concentration. So there's this idea of density dependent motility in active matter, and that's why we looked at that. And even in pitch dark, it's a similar behavior. So then how does slowing down help? Why would slowing down of cells improve the efficiency? The reason being that the reason we think it works is because when you slow down the linear speed, you also slow down the rotational speed. The rotational speed of the cell is linearly proportional to the linear speed of the cells. And so the rotation frequency, so if the cell is turning very slowly, then essentially it's able to average the signal much more over the half revolution, and therefore able to detect the direction of light better and make less errors. So that's the idea. So we can, from these iSpot visualization movies, we can again quantitatively measure the rotational frequency of the cell, and we find that above the critical concentration, it goes down. And so a decrease in linear speed implies a decrease in cell body rotation rate, which enables the cell to collect more photons per unit time and detect the light direction more accurately. Yes?
Expect to see similar phenomenon if you increase the viscosity by...
Yeah, that's exactly the experiment we did.
Okay.
So if this hypothesis is true, then you independently slow down the cells, by fixing the concentration and just increasing the viscosity. So we added some kind of polymer, and we saw that, okay, we can reduce the speed of the cells. And now we are plotting the efficiency as a function of mean speed. So the pink symbols are for data where we have slowed down with methyl cellulose or increasing the viscosity. So we can see that it's actually falling down as we decrease the increase the mean speed, and it nicely overlaps with the data we took by changing the cell concentration. So that completes the story. So, this was an independent validation of our hypothesis that slowing down improves the efficiency of the cells, phototactic efficiency of the cells. And these are my conclusions. I showed you what happens to these cells when you strongly confine them. And I also showed you how they behave collectively during phototaxis. Okay. So thank you.
Hello. Ma'am, in the slide you showed that in the pitch dark,
yes,
at the critical concentration it rises, right? Or
what is rising here?
yeah.
I mean, look at the size of my error bars, right? I wouldn't.
Okay.
worry.
Okay.
Yeah.
Okay.
Yes. What angle? Which angle?
Density, probability density versus radiance angles.
Right.
What is their angle, ma'am?
So okay. I mean, I was plotting it as a function of polar angle. I think you completely missed the point. So yeah, I mean, I can explain here. So essentially, what you would do is that you would draw a circle. So we put the starting point of all the trajectories at 0, 0, and then we draw circles of various radii and ask what is the probability density of this trajectory space as a function of angle. So it will peak up where the light is, right? So that's all. There are 2-3 questions here.
I have one question. So why the cell rotates about itself?
Because it wants to scan its surroundings for light. Otherwise, it has only single eye. If you have single eye, what are you going to do? You have.
So it is not about the hydrodynamic interaction. It is the cell, its own motion.
Yeah, it's its own behavior. It's
And,
a behavior. I mean, that's how the cell works.
Okay, and the MST in the MAST, mean square angular displacement.
Yes.
How do you calculate that theta? I mean, which respect to that particular axis you calculate the theta?
So you are basically, so you have different trajectory points. You are interested in not translational displacement, but angular displacement, right? So you will draw, for example, these vectors, and you will measure the angle between this and this vector for delta t equal to 1. And for delta t equal to 2, you will measure the angle between this vector and that vector. So it's always,
So this is the time series of its trajectory.
Yeah.
So you have xy coordinates and you
join them.
the vector and then calculate that. Okay.
The chemotaxis, like the phototaxis, right? So have you also considered the alignment interactions between two cells, like when they are in the large group, right? Have you considered the idea of including the alignment interactions between the neighbors?
We tried to look for it, and it's not very evident. This cell is not like E. coli, where it will, easily show alignment interaction. So the cell body is more elliptical in nature, whereas E. coli is like a very rod-like shaped object. So it easily goes into these nematic sort of phases, whereas these cells don't. Yes.
instead of pitch darkness,
Yes.
if let's say like when we are shining light from one side, instead of that we shine light from, let's say, two opposite sides,
Yes, yes.
what will be the effect? Like,
So we wanted to do the experiment of orthogonal phototaxis, having one light here and one light perpendicular. And you can ask the question that, let's say you have a population that is oriented towards, so I mean, I don't know what will happen if you have both the stimuli simultaneously on. I think they will just run towards both the sources. I don't know how maybe then you have to really worry about exactly what the gradient is, and maybe more of them will go towards the stronger gradient versus the weaker gradient. I don't know what will happen. But you could also ask what happens when you turn off one and turn on the other one and things like that.
and instead of pitch darkness, let's say if we have uniform light, but some light,
yes,
then will it show similar like motion that it was showing in pitch darkness?
no. So if you just shine blue light on these cells, they will just, attach their flagella to the substrate and sit there happily.
Oh, no motion.
Yeah, I mean, so these flagella are adhesive in nature.
Okay.
So, if it finds truly optimal light conditions, it will just attach to the surface, maybe have gliding motility. So the nature of motility itself changes then. Then it becomes like a walker. Yeah.
Okay.
Yeah. So for a long time, I mean, initially we were novices when we started these experiments. So we started to play around with Clemmy. So we didn't realize that, if you just shine regular white light, they wouldn't move much. And we were like, what's going on? Why aren't they moving?
Okay, nice. Thank you.
Yes, go ahead.
Yeah. Can you please go back to the scheme where you explained the response to the light?
Yeah, sure. It's behind somewhere.
Yeah,
here?
this one.
Yeah.
So in the first step, the eye spot is towards the light, and the cis flagella, the motion suppressed, right? But in the second step, as the eye spot is in shadow region, both the flagella should have equal motility, right?
Not really, because you want to continuously turn the cell towards light.
But
so this is, I don't know, because the eye spot is, so the true picture is very complicated, how the cell is constantly turning towards light.
Okay.
This is more of a cartoonish way to explain what is going on. I don't think that dynamically, I cannot strictly answer your question. You are saying that the two flagella should just be,
yeah, because on the right side you said that in the dark both the flagella move similarly.
No, not really. So in the dark, basically, even if you have dark, the cell will go synchronously. They will move similarly. But then, time to time, there is internal noise in the cell, which would, cause one to beat less than the other. That's how its cell would constantly turn, right? Even in the dark, it's changing direction.
Yeah.
If they keep on beating identical, then the direction will never change.
Right.
So E. coli has to run and tumble, and similarly, these cells also change direction even in pitch dark. Okay.
Okay.
Yeah.
Thank you.
In that case, these flagellas, anyone can move, like maybe.
In absence of a stimulus, anyone can move slightly more than the other, and that would reorient the cell.
Okay.
Okay.
Okay. Is there any other question? No? Okay, then let's thank Prerna one more time.